this lab, we will examine the renal system. First slide is a section in the kidney. Each kidney is covered by connective tissue capsule. This is connective tissue capsule. Each kidney can be divided into an outer cortex. This is the cortex characterized by the presence of renal corpuscles. These round structures are renal corpuscles.
وين الباقيين؟ بس انتوا جايين؟
And medulla is characterized by the presence of tubules without renal corpuscles. بس الميلاد استأذنت. Each nephron has dilated upper part called renal corpuscle found in the cortex. Each corpuscle is composed of tuft of capillaries surrounded by double-walled epithelial capsule called Bowman's capsule. The parietal layer of Bowman capsule is simple squamous epithelium. You can see the nuclei of parietal layer of Bowman capsule. Visceral layer envelopes the capillaries. يعني ما نميزها هنا على السلايد. فقط parietal layer. Between parietal and visceral layer we have a space called renal space. Each corpuscle has two poles. Renal pole, where the proximal convoluted tubule begins, and vascular pole, this is the vascular pole of renal corpuscle. In this region we have complex called JG apparatus or juxtaglomerular apparatus. We can see cells of distal convoluted tubule become modified. We can see the nuclei are directly stained and closely compact together. This region is called macula densa. The cells of distal convoluted tubule modified. We can see the nuclei directly stained and closely compact together. In addition to the JG cells of afferent arteriole form juxtaglomerular apparatus. بس المطلوب فقط الماكيولا دنسا. JG cells ما نميزها بالسلايد تحتاج special stain. This is the macula densa of distal convoluted tubule. This is the vascular pole of renal corpuscle. This is tuft of capillaries, also called glomerulus parietal layer of Bowman capsule, and renal space. In addition to the renal corpuscles, in the cortex, we will examine two types of tubules. First one is the proximal convoluted tubule. The cells of proximal convoluted tubule is very large. The lining epithelium is simple cuboidal epithelium. We can see neurolumen because of the presence of striated or brush border due to the presence of microvilli in the apical surface of each cell. In a cross section, proximal convoluted tubule appear have 3 to 5 cells. In addition, we can see the cytoplasm of each cell are acidophilic cytoplasm. This is proximal convoluted tubule. Distal convoluted tubule cells the cells are smaller than distal convoluted tubule, wider lumen without a brush border or striated border, and lightly staining cells. This is distal convoluted tubule, and this is proximal convoluted tubule. This is another proximal. واضح الفرق؟ Wider lumen without a brush border, more number of cells and smaller cells. This is in the cortex of the kidney. In the medulla, we demonstrate tubules. These are collecting tubules, also lined with simple cuboidal epithelium. These cells stained weakly. This appear lightly staining cells. This is collecting tubules. In the deeper medulla, we have thick and thin segment of Henle's loop. Thick segment is similar in structure to the distal convoluted tubule. Similar in structure to the distal convoluted tubule. This is the thick segment of Henle's loop. Thin segment has a slightly cuboidal or simple squamous epithelium. This is thin segment. We can see the nuclei slightly protrude in the lumen of the segment. This is thin segment, and all these are thick segment, similar in structure to the distal convoluted tubule. This is another thin segment. We can see the rounded nuclei slightly protrude toward the lumen. This is a capillary, smallest blood vessel. In this section, this blood vessel is called vasa recta. This is another one. We can see only one endothelial cells in the lumen. This is another blood vessel or vasa recta. واضح الفرق؟ هذا thick segment. This is the thin segment, and this is capillary. Collecting tubules drain into the collecting duct, and the collecting ducts open at the tips of renal papilla. In the opposite side, we have transitional epithelium. This is transitional epithelium of the minor calyx. واضح؟ Collecting ducts open at the renal papilla, In the opposite side, we have transitional epithelium of the minor calyx. This slide is stained with H&E stain. Second slide is a section in the kidney, but stained with pus stain. Pus stain means periodic acid shift reaction. In this slide, we will demonstrate the medullary rays. Medullary rays are collecting tubules extend from the medulla toward the cortex. هذه بدي تشوفوها، هذه كلها collecting tubules نسميها medullary rays. تمتد من المدلة إلى الكورتكس. نشوف حولها أكو renal corpuscles. إذا جبنا لكم إياها بالامتحان هذه medullary rays والستين هي pus stain. PAS periodic acid shift reaction. Second slide is a cross section in the ureter. The lining epithelium of the ureter is transitional epithelium. Transitional epithelium is modified stratified epithelium. Beneath we have lamina propria. Muscularis layer is composed of inner longitudinal and outer circular. And last layer is the adventitia. Epithelium, transitional epithelium, lamina propria, inner longitudinal and outer circular, and the last layer is the adventitia. The stain here, H&E stain. This slide is a section in the urinary bladder. The epithelium is also transitional epithelium. Cells in the superficial layer. These cells are called dumb cell. The main characteristic feature of these cells are binucleated cells. This is one dumb cell and this is another dumb cell found in the superficial layer of a transitional epithelium. The shape of these cells in the superficial layers differ according to the destination of urinary bladder. Smooth muscle fibers are arranged in different directions. In this section, these bundles of smooth muscle fibers are called the detrusor muscle. يعني ما نميزها، هي المفروض 3 layers بس تكون in different direction. لذلك يعني بالامتحان إذا أشرنا لكم إياها هي detrusor muscle. Last layer is the adventitia with adipose cells and blood vessels. This slide is stained with H&D stain. Last slide, which is a part of endocrine system section in the pituitary gland or hypophysis. Because of its dual origin, hypophysis is divided into two glands, neurohypophysis and adenohypophysis. Neurohypophysis is composed of pars nervosa and infundibulum or neural sulc. The adenohypophysis is composed of three regions: pars distalis, pars intermedia, and pars puberalis that surround the infundibulum. This is pars nervosa, doesn't contain secretory cells. We see unmyelinated nerve axons. The secretion reach this area from the supraoptic and paraventricular nuclei along the axon and accumulate in dilated endings, forming structures called hearing bodies. These structures are hearing bodies. Pars distalis is composed of three types of cells. Chromophils, either acidophile or basophile, according to their affinity to acidic or basic dyes. This is acidophile and this is basophile.
علي هم كافي علي كافي ماما
Chromophobe do not stain in this section or slightly staining. Between the cells we have fenestrated capillaries. Second part of adenohypophysis is the pars intermedia. The cells are arranged in cords or follicles. These cells are slightly basophilic cells arranged in cords or follicles. This slide is stained with H&D stain.